This is one of the major awards of the ACA. The Martin J. Buerger Award is defined as, it's given every 3 years, and it's defined as it's given to a mature scientist who has made a contribution of exceptional distinction in an area of interest to the ACA. So that's a pretty vague description. But in this particular case, it's for chemical informatics. In Helen Berman's case, the award recognizes her contributions to in the pioneering development of information services, which everyone knows that she's associated and is running the protein database, but actually that came after the development of the nucleic acid database. So, okay, I'll try to keep this short. Helen received a AB from Barnard and became part of George Jeffrey's group on carbohydrate structures in Pittsburgh. She remained in Pittsburgh for a few years as a postdoc before going on to Fox Chase, right? And since 1989 has been at Rutgers, where she is now the Regents Professor of Chemistry and the Board of Governors Professor of Chemistry and Biology. She has so many highlights here that I'm not going to have time to go over every single one of them. I'll just point out some of the more significant ones. She's been a member of the USNC, which is a parallel organization of the ACA, from 81 to 93. She was actually the president of the ACA in 1988. She's a fellow of the AAAS in 1996. She won the Distinguished Series Service Award of the Biophysical Society in the year 2000, and this year, 2006, she is the winner, obviously, of the Buerger Award. And among her scientific achievements, she's also been known for her first determination of a single crystal structural model of collagen. And she's also written numerous articles about hydration in nucleic acids and important subjects like that. So with that as an introduction, I'd like Helen to step forward. And I was given this earlier. There's a blank envelope here, which I assume is a check, a valid check. So put this down.
Thank you. So I'd like to thank you very much for this honor and thank everyone who's part of the ACA and helped make this decision. I'd like to thank you very much. And I've been part of this group for a very long time, and that's really what I'm going to tell you about. This is a very personal talk. It's going to trace how I ever got involved in this kind of work and with respect to the protein data bank. It's going to try to put some context into the whole thing in terms of the science of the times that I went through, and then my own science and how it all kind of came together. And I want to do this to kind of give you a sense of how technology and science and community activities all can come together to build important resources that will then enable further science. And so I hope you'll forgive me for the kind of personal nature of this, but I think when you get to be this old, you're allowed to do this. So I'm going to do it. In the 1960s, I will remind you that protein crystallography began to take off. The first structure was determined in 1957 by Kendrew and Perutz, the structure of myoglobin. At the same time, there was an emerging interest in protein folding, and the use of computer graphics to represent structure began to happen at MIT. And the importance of protein crystallography was recognized in 1962 by the award of the Nobel Prize for this achievement. These were the first structures of the proteins that were done in that era in the 60s. Myoglobin and hemoglobin by Perutz and Kendrew, lysozyme in the Phillips lab, and ribonuclease, finally the first American crystal structure. And I remember very, very clearly in 1967 going to the meeting, the ACA meeting in Atlanta, and hearing the presentations of this work by the two groups in Yale and in Buffalo who had determined this important structure. And this is a picture that many of you have seen of the award of the Nobel Prize to the key people who had done the structure of proteins, and then at the same year the Nobel Prize was awarded for the discovery of the structure of DNA. So this was the era in which I was first beginning my life as a scientist. This was the period when protein folding became a very important. Do we have a pointer?
But you have to use the mouse.
Ah, okay.
Because of the fact that we have two screens.
Okay, that's fine. So this is Chris Amundsen, and who's the person who pioneered the idea that proteins will fold and refold. If you denature a protein, it'll come back to its native form. And this is Cyrus Leventhal, who wanted to attack this problem computationally. Now, how does that ever happen? And Cyrus was at the time at MIT and tried to attack this problem computationally. This is the first computer graphics that was used at MIT in Project Mac that Cyrus was so enamored of. And this was the period that I began my scientific career and was very much influenced by this work in my thinking. And so this shows you where I was at the time and what I was thinking about. This is me. I was very skinny and very enthusiastic. This is George Jeffrey, who was my mentor at Pittsburgh. And the work that we did, which is relevant to what I'm going to tell you about, had to do with understanding sugar structures, carbohydrate structures. And my very first paper was in trying to look at a series of sugar structures and understand what was called the anomeric effect in carbohydrates. And these were some structures. What George Jeffrey taught me was the power of looking at structures and looking at groups of structures and trying to analyze them in groups and trying to get some systematics done. At the same time, as part of my PhD thesis, I was on the side rushing up to MIT to try to understand what they were doing up there with respect to protein folding. And my qualifier exam was to create a series of model peptides by looking at protein structures and beginning to see whether they had common features and to analyze the sequence structure relationships, synthesize these peptides and understand their structures. And this all came out of hanging out in Leventhal's lab and trying to, and so this was sort of a dream that I had, that we'd be able to do something like that. In order to do that dream, you had to have the coordinates of the structures. And of course, to get coordinates of protein structures back in the 60s was not an easy thing to do. They appeared on, the structures were on computer punched cards. So there was a grassroots effort by the community to try to archive data, and not surprisingly, I was part of that grassroots effort. At the same time, which means that I just tried to make a lot of trouble and get people to somehow put these data into one place so that the rest of us could use them. The protein crystallographers began to discuss how to best archive the data, because they began to realize that they had something very important, and this very important data was really, really hard to ship around the world. Every time some graduate student or postdoc said, I'd like to have the coordinates of myoglobin, what were the people supposed to do to get those coordinates to them? We were not in any way in the internet era. So in 1971, there was a meeting at Cold Spring Harbor at which these two groups of people were present, the people who had determined these structures and knew it was very important to get the data out to the public, and then people like myself who crashed the meeting, who were not invited to the meeting, who showed up in Joel Sussman's Peugeot, deciding that we had to somehow get these data. And we got together at that meeting, and we talked to Walter Hamilton, who appeared from Brookhaven, who also decided he had to find out what was going on. And we convinced Walter that this was very important. At the time, Edgar Meyer was spending the summer in Walter's lab and trying to make a protein library. And Walter said, okay, we'll make a protein data bank. He then went to Europe and he talked to Olga Kennard, because he recognized that protein crystallography in all science is international, and you can't do these things unless you get international agreement. And so in October so the meeting was in June of 1971. In October of 1971, the PDB was announced in Nature New Biology, which was one of the Nature journals at the time. All this fuss over 7 structures, that's what we were talking about. In 1975, PDB received its first funding from NSF. For those of you who might be interested, that was for $33,000, and at that time there were about 32 structures. This is the era that we were living in. This is the PDP-11 computer. This is a CDC 6600, which was at the time, the supercomputer of the era. This is a Richards box where people were fitting their protein data maps with this humongous setup that you had to have a very big room to do this work in. And then this is a Vector General graphics computer, which was one of the first computers that was used for the purpose of fitting electron density data. And this is the famous punch cards that we all had to live with back then. As I told you, there were people who thought that the only way you could get anything done, remember this is in the this is in the era of student protest and student power, and everything we did involved writing petitions and doing things like that. And so here's the petition asking somebody or other, and I'm not really sure who we were asking, to give us a protein data bank. And this petition was done in about 1970. And you can see there I am with a handwriting that you can actually read. This is Ned Seaman. This is Don Vogt. Howard, you're here. Howard Einspar, and so on. And so we all thought this was really important. This is a picture of Walter Hamilton talking to Hal Wyckoff at that meeting in 1971 in Cold Spring Harbor. And this is a picture of Walter and myself and Tom Ketzel, who took over the protein data bank after Walter's death, on a boat somewhere in the middle of Europe going to a meeting in Denmark. And this is the article about announcing the protein data bank. So here's where we were in the 70s. There were, in 1979, a little over 100 structures. These are the type of structures that were in the protein data bank. The protein data bank at that time was at Brookhaven National Lab. Tom Ketzel was the person who was in charge of that project, and Francis Bernstein was the one and only curator of the data at that time. And And what was I doing at that time in the 70s? I was at that time interested in nucleic acid structure and in the systematics, and this is that structure of UPA that I worked on with Joel Sussman and Ned Seaman and Sung-Ho Kim. And this is a structure of structures of nucleic acid drug complexes that I worked on with Steve Needle. Here's a picture of Steve and me, and I'm looking very intense. This is in his office in London. And again, the whole idea here was systematics. How do we look at the systematics of structure and understand structure, not just one at a time, but groups at a time? So this was part of the way I was thinking then and continue to think now. So that was the 70s. That was the beginning of the Protein Data Bank, and the idea that you could, by collecting data, you could somehow do systematics of structure. In the 1980s, the technology began to take off for doing structure, and the technology in every single area. So for example, in molecular biology, it was made it possible to get large quantities of material. That had not been possible in the old days, as many of you know better than I know how difficult it was to get enough material to do a protein structure. The instrumentation for doing, structures in terms of the data collection also began to improve with the advent of synchrotrons. The computer hardware and software for doing our kind of work began to evolve. And so the technology for doing this kind of work began to improve. So that made it possible for structural, I'm afraid to touch this. Oh, God. Okay, I got it. Structural biology then began to focus on medical problems. So instead of working on structures because you could crystallize them, you could actually pose a medical problem and begin to do the work. A very important thing that happened in that era is that community effort to promote data sharing began to ramp up. And this was as a direct consequence of Marvin Kastman, who was at the NIH at the time, said this was the beginning of the AIDS epidemic. We didn't know it was going to be an epidemic, but we thought it was going to be an epidemic. And Marvin wanted to fund people to do AIDS research, and he said, I'm not going to fund this kind of work unless I can have some assurance that the data that are produced, structural data, are out in the public. And he made a lot of phone calls to different people in the community, including myself, saying we've got to make some kind of policy to make sure the data are publicly available. And so there began a series of community efforts to promote data sharing, and that ended in 1989 by the publication by the IUCR of a set of requirements, guidelines for requiring data deposition. So here we are in the 80s, being able to get beautiful crystals relatively, compared to before, relatively rapidly. Here are some of the key players. Of course, the ability to have synchrotron radiation, and of course at Brookhaven was one of the leading places at that time. Wayne Hendrickson is a key person in all of this because Wayne developed methods using MAD to phase protein structures. Alwyn Jones is another very important person in this era because he was able to write the program that would allow you to fit the electron density on a computer instead of with the Richards box, and then of course the ability to have all of this instrumentation. So let's talk a little bit about this cooperative community action and how that worked. There were letters to the editors of journals by all kinds of people, including myself, And there were at least 3 different committees that were set up to do this work. The IUCR Commission on Biological Macromolecules was the most official committee, let's say, that was chartered to look into how to do this. The ACA and the USA National Committee also set up a group to look into how to do data sharing. And then Fred Richards was a tremendous pioneer in this area, and he made phone calls and also took the petition route for being able to get people to think hard about getting their data out to the public. The funding agencies were sort of in the background. They wanted this to happen, but at least in America, the way it works with the funding agencies is they will not do anything unless the people they perceive as the community tell them that this is the way things should be. And so it was up to us as a community to figure out how to regulate ourselves. And I have to say that we're, as a crystallographic community, structural biology community is one of the most active and forward-looking in this area, even though some of you may know that we've been accused of, quote, holding stuff back. Compared to other communities, we really tried to take care of our issues having to do with data sharing relatively early in the game. And then there were articles in journals. So I'll walk you through some of this. So this was a letter that I wrote in 1986, and I wrote it to Dan Koshland, who was the editor of Science at the time, and I said that the data belongs at Brookhaven. It has to be submitted. And I suggest that if he needs more advice, he ought to talk to Michael Rossman, who was at the very beginning involved in all of this and continues to this very day to be a tremendous advocate and leader in the area of data sharing in structural biology. And then John Norvell, who is still a very active person in the funding agencies at NIH, wrote a letter saying it is NIH policy that results of the sponsored research should be published and made available. And so he's saying he's very happy to hear that there are these committees being set up. This is a letter from Charlie saying that there would be an ACA committee to set up standards for data sharing. And then another letter from Wayne Hendrickson saying that there would be a meeting of this IUCR committee at Perth, which was in 1987. So there's lots of activity in many directions, as shown here. These are all the sort of key people who were involved in the setting up of these standards, including Eleanor Dotson, Mike James, Brian Matthews, and so on. So this was the this was the IUCR Commission on Biological Macromolecules. The Bug Committee had myself on it, and Wayne, and Michael James and Tom Ketzel as the head of the PDB. And then the Richards Committee had as the key people Michael Rossman, Dick Dickerson, and the Richardsons. And then there were all the, and then the people involved in the funding agencies who were being kept in the loop were John Norvell and Marvin Kastman. And then Art Kowalski was the person at NSF in charge of the funding for the PDB at the time. And this is the petition, a much more formal-looking petition that Fred Richards put together, in which he calls for the importance of doing data sharing. And this petition was signed by a very, very large number of people. And so all of this came together. So this was a process that took several years, and it all came together into this document that talks about how you should do data sharing. And this is the document that it was published in ACTA-CRIST and is still what we has the rules that we live by. Some of it has been modified in the sense that we now release data sooner, there are fewer holds and things like that, but the spirit of this document is such that it's the one that we live by. And so by the end of the 80s, we start to see two trends. One is the number of structures begins to increase even more, but please look that in 1989 there were still only 500 structures in the PDB, and as you all know, there's about 38,000 structures in the PDB now. The kinds of structures that were in the PDB and began to be put in the PDB changed because the science changed. And so in the early days we had these very nice small enzymes, and by the mid-80s we start to see the rhinovirus structure, we start to see DNA polymerase, and we see the beginnings of nucleic acid crystallography where the structures are much smaller. It took a long time before that work actually took off. And again, we start to see a lot of these structures coming in to the PDB. Okay, so this is, what was I doing in this period? What I was doing, again, was systematics. I wanted to understand the systematics of nucleic acid structure. And I did this work with Stephen Needle, my very close colleague and collaborator. This beautiful picture was, I think, drawn by Stephen, showing the hydration of a small dinucleoside phosphate drug complex. And then from that, I start looking at hydration around bases in nucleic acids and begin to discover that, in fact, it's much more orderly than we had previously thought. And then this is a structure of actinomycin with a dinucleoside phosphate that was done with Fusao Takasugawa. And the other thing that was happening to me in the 80s is I was raising my son, who I have to embarrass here. He's sitting in the back of the room. And this is a picture that he drew when he was a little boy, where he somehow knew that cells go to DNA, that he understood the concept of scale, and he has cell DNA, and then he has this picture here, and he winds up with quarks. And he told me, and he said he didn't want me to show this picture because it's wrong, but what do I know about quarks? But anyhow, that was an important part of my life in the 80s. So what happened in the 90s? The number of structures began to increase exponentially. The complexity of the structures also began to increase. And then new databases began to emerge, such as the nucleic acid database, which was something that I began to work on in the beginning of the 90s, and that was completely motivated by the idea that I really wanted to be able to do something with groups of structures in a much easier way than I could do with the data as it was organized in the PDB at the time. And at the time I felt that nucleic acids were being treated as stepchildren in the macromolecular community, and I wanted to do something about it. And so that's how I got involved in the NDB, and it came directly out of my own research interests in nucleic acids. The user community for the Protein Data Bank began to expand dramatically. The PDB used to be a place where people put data, such like in a storage box on Route 1 in New Jersey. They just kept it for safe keeping. The idea that somebody else would analyze your structure was still something that many people did not like. But in the 90s, people began to do that, began to analyze people's work and people's structures and come up with new science. At the same time, the mmCIF dictionary was created, and you'll hear a little bit about that today. And that was done, my interest in that was the need if you're going to have a database, you've got to organize your data in a very, very rigorous way and have very rigorous definitions for everything. Otherwise, it's impossible to be able to get anything out of the database. You can pour it in, but you can't get it out. So that was the interest in mmCIF. And then in the 1990s, the PDB moved, after 27 years at Brookhaven, it moved to the RCSB, which was a consortium consisting of Rutgers, San Diego, UCSD, and NIST. And that was as a result of a competitive bid that was made in 1998. So here's a picture to embarrass Paula and John and Phil Bourne and Sid Hall and Shoshana, and this was one of Shoshana Vodak, and this is one of the early CIF meetings in Brussels, which was a major breakthrough in how to organize the data. And I don't know whether this is before or after the champagne, but that was a very, very important meeting. Here's the CIF page from the PDB, and there's lots and lots of resources now for mmCIF that we maintain. This was an ACA newsletter from 1997, which focused on all the different kinds of databases that had begun to emerge during this period of the 90s, where people wanted to look at specific aspects of structure, and there were many different databases, and people began to experiment with this. And so by the end of the 90s, we had something in the order of 10,000 structures in the PDB. Okay, and at that time in my research life was the NDB. This is the original homepage from the NDB, which was our attempt to organize nucleic acid structures into a relational database. This was a sort of culmination of the work that we did in hydration, where we were able, in collaboration with Bodan Schneider, we were able to see that the hydration nucleic acids is very predictable. I mean, you can take a nucleic acid structure and using the systematics that we had worked out, you can predict exactly where the waters are. And then this is work on collagen structure, which you'll hear about perhaps, but this is work that was done with Jordi Bella, where again we began to see it's not just DNA where you can understand the hydration very well, but also collagen. And then we also did work on cap DNA complexes, and again with that we did about 15 of these type structures, and we're able to do again the systematics of this particular system. And then this is my research group at that point in the 90s, and here I am weighing a lot more than I did when I was at the picnic in Pittsburgh in the 60s. And this was the group. I think this is you, Jordi, and Gary Parkinson, among others who were in the group. Okay, and so now we're up to the 2000s, and there's this continued growth in structures, and now we're really getting overwhelmed. There's so many structures, and structural genomics begins to take off, which is this attempt to look at structures on a genomic scale, and the formation of a formal organization that formally recognizes that the PDB has to be a global organization, so this is called the Worldwide PDB, and this is to ensure that the structures are never sort of broken on any kind of regional lines, and that the data stay uniform. Structural genomics, as many of you know, is the next step beyond the Human Genome Project, and the concept is to do structures on a genomic scale, and this is work now that's in its second phase, the production phase, and many of the people in this audience are involved in this particular effort. This is a slide that was made in September of 2005, which basically has all the structures up to that point that had been done in this high throughput genomic way in the structural genomics projects in America. The Worldwide PDB now consists of 4 organizations. In addition to the RCSB group, there's the PDBJ, and I was very happy to see Masami Kuzunoki here from that group. The MSD group, which is at EBI, and now Biomag ResBank is now also a member of WWPDB, because they soon will be issuing IDs for structures that are coming in through their site that have coordinates. And so anybody who issues a PDB ID must be part of this organization and follow the rules that we've made up. And then this is a picture of 3 of the members, myself, Haruki Nakamura, and Kim Henrik, who are the founding members of WWPDB. So WWPDB has a set of rules. A lot of people, nobody ever said that America owns the data. Maybe George Bush thinks we own the data, but we don't own the data. And anybody can go and take these data, especially because we've made them completely freely available, and just compete against us. And that may not seem so dangerous until you start thinking about picking up a hemoglobin file from one place and getting a completely different set of coordinates than from another. So we felt we had to set up a set of guidelines and make sure that the data would always be reliable, and a hemoglobin file is a hemoglobin file is a hemoglobin file. So that's the idea of WWPDB. And we have now yearly meetings and various conference calls to make sure we keep this, keep the archive single. So now we're up to almost 40,000 data. When we took over the PDB management in the year 2000, we began to see the first ribosomal subunit structures come in, and that was an enormous challenge to see structures that complex coming in. And I could only sort of thank God that we had been knowing how to handle nucleic acid structures from our NDB work, and we were able to process these data, which are so much more complicated to handle than, say, myoglobin. Somehow this did not come out right. Oh, there it is. Okay. Don't know what happened. Okay, now in this era that we're in now, we're starting to see structures coming in not only by X-ray and NMR, but they are coming in from cryo-electron microscopy. And so we are forced to think about how to represent structures like this that may not necessarily have coordinates. And so this is a picture of the increase of data using structures that are using electron microscopy, and here's Hua Chu, with whom we've been collaborating to come up with a way to handle these data properly. And one of the lessons that we learned in handling our data is that we must not treat X-ray data, NMR data, and now cryo-EM data the same way. They have their own special attributes, and we need to understand the domain really well. And so that's one of the things that we spend a lot of time on in the PDB project. And so what was going on in my lab in the 2000s is mostly looking at larger complexes related to cap DNA and different pieces of RNA polymerase, and then looking at the systematics of the cyclic AMP binding to different kinds of proteins. And again, systematics is really key to what drives the work in my lab. And this is a picture of the group that had worked in this area. And Kathy Lawson became a part of the group several years ago when she moved from Brookhaven to Rutgers at the same time as the PDB did, but for different reasons. And now Kathy has taken over all of the experimental work in this area, and I no longer am doing experimental work at all in my group, and Kathy is leading this project. So what has the PDB enabled? Minimally safe storage of protein data. So some of you have called up and said, help, my data is in the dumpster in the back of the chemistry building, and I didn't back anything up, and so but we do, and so we can give you the data. There are molecular replacement models for structure determination, parts list for modeling. This is especially important for cryo-EM. Structure-based drug design, PDB is crucial for that. protein structure classification and protein structure prediction are all things that the PDB has enabled. And so here are some of the resources that have evolved because the PDB is there with the data. There are other resources that now can pull the data from the PDB and do different kinds of classification. And so we have CAF and we have SCOP, which are extremely important databases that classify the protein domains. There's the effort to predict protein structure that's done by CASP, and CAPRI is a similar effort to CASP in which the attempt is made to predict the protein-protein interactions. And we, as in the PDB, have worked closely with these groups to make sure that the sequences are available as soon as possible before the publication of the actual structure. Okay, the PDB is now very heavily used. The primary citation that we have, that when we took over in 1998, is an article in Nucleic Acid Research Database issue, and as of a couple months ago or a month ago, there were 3,900 citations, and that's a lot of citations. And because I wanted to see what it is that people are citing us for, I looked at some of the articles, and there's a huge number of articles of people trying to figure out how to do, for example, protein prediction, the thing that so captured my interest back when I was a student, and all sorts of other analyses of proteins. So the PDB has become much more than a storage facility for protein structure. So what would the world be like without the PDB? Suppose you needed information about a single structure and there was no PDB. You would read the article and you'd say, gee, that looks really interesting. You'd contact the author to get coordinates. And if you were lucky and the author was really, really cooperative, in maybe 2 weeks, 4 weeks, depending on the author, you could get your analysis going. Supposing you wanted to do structure classification, and again, no PDB. You'd look up the articles, you'd contact the many authors, you'd somehow figure out how to put the files in some kind of format that you could work with, and maybe it would take you a year or two to begin the analyses. Some of you might think about Google and maybe how that might be changing, but this is right now. And supposing you wanted to do structure prediction, and that is you wanted to take what's known, you do a knowledge-based structure prediction, and there was no PDB. This is a sign as you leave Long Island towards Brooklyn, and you're leaving Brooklyn, there's a sign that says forget about it. That is, forget about it. That's a real sign. The president of Brooklyn, where I come from, thinks the same way as I do, and I would, if I was the head of Brooklyn, I would have that sign too. So so anyhow, that is in my view of what would happen if you wanted to do structure prediction and there was no PDB. So that's a kind of walk through the world in which I lived as the PDB was growing up and the influences that were on me. And this, I first of all like to thank my students and postdocs and collaborators from my days at Pitt, which was really important, Fox Chase and Rutgers, and all of my colleagues and friends in the ever-enlarging worldwide PDB community. And here's a picture of the PDB group in 2005. We have a retreat every year. Phil Bourne and John Westbrook and have been with the PDB since the RCSB PDB since 1998. Kyle Burkhardt is the lead annotator, and many of you have corresponded with Kyle, and she's a key person, and Zukang Feng is the person who's written the software that lets us process the data relatively rapidly. And there's all these people, and they're all really important. And the other group that's been super important to us in running the PDB in the last 8 years has been our advisory committee. We have an international advisory committee, and Steve Burley has been the head of that committee from day one. And this advisory committee, while they always tell me they work for me, they're tough on us, and they tell us when we do things wrong, and they tell us when we do things right, and they're always presenting challenges about what we should be thinking about in the future. And this is a picture from the last advisory, which was about a month ago in California, and I particularly want to thank this group for being right on in their criticisms of what we're doing. And I'd finally like to thank all of you for hearing the story, and I would invite any questions if you would like to ask them. Thank you.